Hi there, Saphira and I are here today. We're going to talk to you some more about cellular structure and function, and today we're going to talk about cellular transport. All right, this is really important for helping organisms such as Saphira and ourselves maintain homeostasis. All right, so we've got to be able to bring important nutrients into the cell, but also get rid of waste, and we're going to talk about how your body needs to do that. All right, so let's get started, and we will discuss these tomorrow. All right, so cellular transport is the movement of substances either within the cell or it can be even into and out of the cell. All right, there are two main categories. There is passive transport, which is the movement of substances across a membrane. This requires no energy because it moves with the concentration gradient. All right, concentration gradients are set up when there is more of a substance either inside a cell or outside the cell. All right, so in passive transport, things are going to move with the concentration gradient from an area of high to low concentration. Active transport requires energy because it's going to move against this concentration gradient. We're going to actually have to pump molecules across a membrane from an area of low to high. So it's going against the concentration gradient. Some examples of this are solute pumps, exocytosis, and endocytosis. All right, so the first category, which is part of the passive transport, is diffusion. So diffusion, very simply, is the net movement of particles from an area of high concentration to an area of low concentration. All right, and if you look at this diagram here, we are adding dye to water. All right, in the beginning, there's a concentration of high dye molecules on this side, and then once we let it sit for a while, they start to spread out until equilibrium is achieved. All right, and equilibrium means that there is a uniform mixture of those dye particles throughout the solution. All right, this is actually what we want to achieve is dynamic equilibrium, which means that the concentration is going to stay the same, but because the particles are continuously moving, the movement is continuous, but no change in concentration is going to occur. All right, this type of movement, which we call diffusion, requires no energy. All right, facilitated diffusion occurs when we have rather large molecules that need to move and they cannot pass through the phospholipid bilayer. All right, so we use these channel proteins. If you remember from the handout that we had, these channel proteins can span the entire phospholipid bilayer, and ions or molecules can move through the center of them. All right, no energy is required because we're still moving from an area of high concentration to an area of lower concentration. All right, so this is the way that things would naturally move. So facilitated diffusion just requires a transport protein. All right, now we are talking about osmosis. Osmosis is very specifically the diffusion of water and water only. All right, if you remember, water is a solvent in a cell, and it will diffuse toward the side of the membrane with a greater concentration. All right, if we have a membrane and the solutes, which are the particles, for example, salt, and those particles can't move across the membrane, in order to achieve the same concentration on either side of that membrane, water will move. So you notice over here in this diagram, since there's a higher concentration of particles, solute, on this side of this membrane, all right, those particles can't move, so water must move to balance it out. All right, so we see an increase of water. That way we have the same concentration. So a very simple rule to remember when we're thinking of this is just very simply that salt sucks. Salt is a solute, and it will draw water in its direction. This is why you get a signal from your body, hey, you're thirsty, especially after you eat something salty. It's because those cells in that immediate area, in order to offset the amount of salt in your system, it actually pulls water out of your cells. Then that sends a message to your body saying, hey, we need to replace this water. So it sends a message to you. Right, tonicity. Tonicity is the ability of a solution to cause a cell to gain or lose water. All right, so this is what I'm talking about. The idea that that salt would suck water out of your cells. All right, the solution that the cells are in has a certain tonicity. So it depends on the concentration of solutes inside the cell as well as outside of the cell. Typically, these solutes cannot move across the membrane, so water has to move. All right, this is crucial to the function of all plants and animals, because if you lose too much water, it can be detrimental, as well as if your cells bring in too much water, it can cause the cells to swell and even burst if they're animal cells. All right, so we're going to talk about 3 different tonicities. The first one that we're going to talk about is an isotonic solution. Iso means the same. So in an isotonic solution, it's a solution that has the same concentration of water and solutes. So solutes could include sugars, proteins, any other substances. All right, it's the same concentration on both sides of the membrane. So water will continue to leave the cell, but it leaves and enters at the same rate. So it doesn't change the concentration on either side of the cell. So the cell will retain their normal shape. All right, so if this is a cell here, this is the plasma membrane of the cell, and there's about a 10% salt concentration inside and outside, water will continue to move because of that dynamic equilibrium that we have, but it will move in and out at the same rate. All right, now let's take a look when we talk about a hypotonic. Hypo is when the concentration of solutes is less on the outside of the cell than on the inside of the cell. So if you take a look, there is actually more water outside, more solutes inside. So the net movement of water is going to be into the cell. Remember, we said salt sucks. So wherever the higher salt concentration, that's where water is going to be pulled. So water in this situation would move into the cell until we got a 10% salt solution inside the cell. And then dynamic equilibrium would occur. The problem with a hypotonic solution is that if, especially for our animal cells, which do not have a cell wall, if the osmotic pressure inside the cell increases too much, it can cause the cell to burst. So animal cells will swell and could burst. Plant cells will swell and push against the cell wall, making them firmer, but they would not burst because that cell wall helps to protect them. All right, and lastly, a hypertonic solution means that the concentration of solutes outside the cell is much higher. So in this situation, the salt concentration inside the cell is 10%, whereas the salt concentration outside the cell is 20%. So water is going to be pulled outside the cell, and if for animal cells, they'll shrivel up, and plant cells will wilt and shrink away from the cell wall as a result of this. So if we have solutions that are too hypo or hypertonic, then we can have a lot of bad conditions occur in the cell. For example, this is showing you on the top animal cells, red blood cells, and then on the bottom we see plant cells. So in a hypotonic solution, if the concentration inside the cell is too great, then it can cause lysis, or the bursting of cells. For plants, it just causes them to be really firm. And then we look at an isotonic solution. This is the way cells should look. Hypertonic solution, the red blood cells shrivel up, makes it hard for them to do their job. And then also for the plant cells, the actual inside cell pulls away from the cell wall, and that can cause problems for the plants as well. Oftentimes, you'll see wilting in the plant. All right, so back to our different movement of molecules. So that information was passive transport. Now we're going to talk about active transport, which is the movement of substances against the concentration gradient. The key thing to remember here is that it requires energy. So we have to have these pumps. All right, this is showing you the pump here. And the pump is basically ATP. In this case, it's called the ATP ACE pump. So ATP is an energy carrier in your cells. We're going to talk a lot about this in the upcoming unit. All right, ATP is the primary energy carrier in your cells. So we're going to spend a little bit of that ATP in order to force these solute particles from an area of low to high concentration. All right, notice we have a protein here, but rather than just a simple carrier protein, it's actually going to be called a pump because we're going to actually have to use energy to pump the solutes across the cellular membrane. All right, other types of active transport includes endocytosis. If we have particles that are much too large or to enter the cell, then they can get embedded in part of the plasma membrane and it'll form a vesicle around them, and then that will pinch off and allow these particles to enter the cell. All right, so this can either be diffusion, so if we're going from an area of high to low, or it can be active transport in which it would require energy. So endocytosis can be included in either both active or passive transport. It just depends on the concentration gradient. But the important thing to remember is that these particles are too large to pass through a carrier protein, so they kind of embed themselves in the plasma membrane. That pinches off and will become a little vesicle within the cell. All right, exocytosis is the very opposite. It's when we need to get rid of rather large particles that cannot exit through one of those channel proteins. So they will embed themselves in the plasma membrane, they will come to the surface, and then they will be expelled out into the aqueous environment of your body. All right, so this is how we're able to get rid of waste products in the cells, and this is also how the cell releases hormones to act as enzymes to carry out other reactions in other nearby cells. All right, so there's a lot of terms in this section. Please make sure you complete your notes, bring them with you tomorrow so we can discuss them in class. Have a great day.